glycogenolysis, so breakdown of glycogen, is also stimulated. It's a bit of a unusual word there. I'm not that familiar with it, but this means breaking down glycogen. Okay? So then in muscle, we have epinephrine, right? Epinephrine also works on liver, but when it works on liver, it has these effects. Right? When it works on muscle, it also breaks down glycogen to glucose 6-phosphate, as here, right? But we know that muscle does not export blood glucose, right? And also muscle does not have a gluconeogenesis pathway. Right? It just doesn't have the enzymes. But then in liver, glycolysis rate is cut way down so that the glucose can be exported. Whereas in muscle, the glycolysis rate is actually increased because the muscle is the tissue that needs to do the work. Right? So then the question becomes, you know, what's up here? What's up with this difference in glycolysis? So how can glycolysis go up in muscle in response to epinephrine when it goes down in liver in response to epinephrine? And so the answer to that comes from this slide here, which we looked at before. In the regulation of pyruvate kinase, the last enzyme of glycolysis, it says pyruvate kinase L/M, liver/muscle, to distinguish those two isozymes, right? And then here it says liver only. So there are, whatever, 5 or 10 or 20 different amino acids differences between the protein that's made in liver, that's pyruvate kinase, and the pyruvate kinase that's made in muscle. And the liver pyruvate kinase isozyme, when PKA is around, right, when it's been activated by the upstream triggers, which come from glucagon, it will phosphorylate pyruvate kinase in liver and thereby inactivate it, and thereby turn off glycolysis in liver. And so the distinction is simply that in muscle, that particular pyruvate kinase is a different isozyme, and it's just not a trigger. It doesn't interact. So muscle pyruvate kinase does not interact with PKA, does not get phosphorylated and therefore inactivated. So if that happens, then here PKA goes off and phosphorylates pyruvate kinase, going off to the right, but it also activates this cascade here. So this breakdown of glycogen is still preserved, which is what we want in muscle. We just don't want to run glycolysis as well. Does that make sense? So there's two isozymes of pyruvate kinase that are present in this, for example, in the human genome. And when stem cells differentiate, and I don't know the whole pathway of it, but eventually you get to a liver cell and you get to a muscle cell, right? In the liver cell, the muscle isozyme, which is sitting there in the DNA somewhere, is bound up in the middle of the chromatin with nucleosomes piled all over it, and then completely inactive, right? It's not getting transcribed. Right? Whereas in muscle cells, it's the liver isozyme gene that's not getting transcribed. Okay. Questions? Okay. So let's move on. So that's the end of regulation and the beginning of nitrogen. So we don't spend a lot of time in the class on nitrogen. We'll be looking towards the end of this at this interplay of these different cycles. The TCA cycle, of course, you already know about. We have dietary protein, we have intracellular protein getting broken down, and in both cases we get amino acids. And we divide conceptually, we divide the amino acids into the nitrogen, which is just the alpha amino group, and then the rest of it, which is the carbon skeleton. Carbon skeletons of amino acids are converted to alpha keto acids, which feed into the TCA cycle and can get oxidized for energy. But it's not very common, right? So if there's a lot of protein turnover for some reason, if you eat a ton of protein, or if you're starving, you can oxidize your amino acids acids take their carbon skeletons into the TCA cycle and actually burn them. But typically, it's fats and sugars that are burned, not amino acids. There's no way to store amino acids, so the body has to do something with them. Typically, the process is to recycle the nitrogen, to recycle the skeletons and to excrete the nitrogen as part of the turnover process. And so what we're really looking at today is how does that nitrogen get excreted, right? Which is the function of the urea cycle. Okay. So we're going to talk about, so this is just proteases in the digestive tract, right, or else turnover of protein inside the cells. So what we're going to talk about is the release of the ammonium, right? It can get incorporated in anabolic pathways as well, which we're not talking about. So that's one thing we're not talking about. The ammonium is converted into a molecule called carbon monophosphate, which then goes through the urea cycle, which has a link to the TCA cycle. Okay. The carbon skeleton part of the amino acid, there's a whole array of pathways here to take the carbon skeletons into compounds that can feed into the TCA cycle. And so we're not considering any of that. And so there's a lot of pathways, there's a lot of interesting chemistry. We just don't have time to get into it. This is giving you a summary of it. So TCA cycle. The different amino acids here, either in pink or in blue, are feeding into the TCA cycle at various places. So if they feed in later in the cycle and they sort of bypass where the ATP generation is, like there's NADH forming steps, at least one early in the cycle over here. So if you feed in here, then you wouldn't get that ATP. Right? So we've got pink ones are called glucogenics because it's possible to run that carbon back through gluconeogenesis and make glucose out of it. So if it's theoretically possible to do that, then we classify the amino acid as glucogenic. On the other hand, some amino acids are both glucogenic and ketogenic, and a couple of them are exclusively ketogenic. The ketogenic amino acid is one that gets processed in the liver into compounds that are called ketone bodies, one of which is acetone that just gets exhaled. Another one is acetoacetate, and another one I think is beta-hydroxybutyrate. I'll have it on the slide on Wednesday. But those ketone bodies in the liver get exported through the bloodstream to other organs, and then they get used metabolically to provide energy. Like they feed into TCA cycles in the other organs. So that's another possible breakdown pathway for amino acids, but only some of them go that way. And some of them go both ways. Like for example, isoleucine is here, isoleucine is here. So it's possible for isoleucine to, depending on what enzymes are working on it and what quantity, it could go one way or the other. Yeah?
Should we feel or recognize which are glucogenic, or would you indicate that?
No, I mean, I wouldn't expect you to remember, the details. The only detail of conversion will be the transamination reactions, which we'll talk about. We've mentioned already, and I'm going to talk about now. Right. Otherwise, don't worry about memorizing which amino acids are blue and which ones are red here. So that's so crucial. Okay. This slide I've annotated this a little bit this morning, so it's a little bit different than what I have online. But what we have in amino acid catabolism, so we've got amino acids from ingested proteins. Here's the amino acid, right? And the key one of the important key reactions is this one with the transaminases. And the transaminases function, so the amino acids that come in from cellular breakdown or through the diet, all of the nitrogen ends up getting shuttled into glutamate first. Okay, use glutamate, and then glutamate is broken down by this enzyme, glutamate dehydrogenase, to yield ammonia, which can go through the, or urea, that can go through the urea cycle. Okay? But before that happens, right, so that would be this reaction here, glutamate dehydrogenase is this arrow here. So before that happens, we have transaminations for amino acids coming in from cellular protein and turnover. We have transaminations for alanine coming in from the skeletal muscle. That's another important source. And so these are liver reactions here now. The liver is a big metabolic organ. Right? So whenever you have a transamination, so here's one and here's another one, the way they work is there's always two substrates and two products. And it's a ping pong kind of mechanism. Okay? And it always works with a specific cofactor, which is called pyridoxal phosphate, PLP, which we haven't encountered yet. Okay? And so as I mentioned before, the way the transamination reaction works is if you take an amino acid, an alpha amino acid, and you just take the alpha carbon and convert it to a carbonyl group, then you've got an alpha keto acid, and that's the reaction that's catalyzed. And what I did not say before was that as one of these reactions happens, if alpha Kg is converted to glue, that's an alpha keto acid going to an amino acid. At the same time, a different amino acid is going to an alpha keto acid. Some transaminases are very specific for which particular amino acids they will work on, and the transaminase enzymes, they're also called aminotransferase enzymes, are named after the amino acid part